Veterinary virology. Welcome. Let us read this photo. Let us read this photo. Infectious disease is one of the few genus and nature left in the world. The dangerous are all dead, and the land grows rusty in the chimney corner. About the only sporting proposition that remains unimpaired by the relentless domestication of once free-living human species in the war against those precious little flu creatures which lurk in the dark corners and stalk us in the bodies of rats, mice, and all kinds of domestic animals which fly in the cradle with the insects and wailing us in our food and drink and even in our love. This photo is taken from the book Rats, Lice and History, written in 1935 by the great microbiologist Hans Zenzer, as he reflected in his life in infectious disease science. Let us put a definition of the virus. Simply, what is virus? Virus may be defined as a cellular organism whose genome consists of nucleic acid and which obligately replicate inside host cells using host metabolic machinery to different extent to form a pool of components which assemble into particles called virion. Viruses can't be grown on sterile media but require the presence of specific host cells. A virus differs from a cell in three fundamental ways. A virus usually has only a single type of nucleic acid serving as its genetic material. This can be single or double-stranded DNA or RNA. Viruses contain no enzyme of energy metabolism, thus can't make ATP. Viruses don't encode sufficient enzymatic machinery to synthesize their component macronucleus, specifically no protein synthesis machinery. Fundamentally, then a virus is a package of genetic information protected by a protein shell for delivery into a host cell to be expressed and replicated. Viruses are distinguished from other obligate parasites, some of which are even simpler than virus, mycoplasma, small bacterium that grows only in complex media and attached to eukaryotic cells, chlamydia, obligate intracellular bacterial parasite which depend on eukaryotic cells for energy, protozoa, obligate intracellular parasite that replicate within eukaryotic cells, varioid, infectious agent of plant that exist as naked nucleic acid, circular single-stranded RNA. Hepatitis delta virus, HDV, viroid-like agent whose replication is dependent on HB hepatitis B virus. And this is Stanley Berliner, the winner of the Nobel Prize in 1997 for his discovery of barium. Barium, proteinaceous infectious agent, hypothesized the identity of the unconventional slow virus such as Corus, Crady, and mad cow disease agent. No nucleic acid is known to be required for barium function. They are thought by many to consist slowly of protein and perhaps diseases caused by barium or barium diseases, Creutzfeldt-Jakob variant, Creutzfeldt-Jakob, Straussler and Schenker syndrome, fetal familial insomnia, and corona. And this is the histopathology of some of these diseases: Crady, PSA, bovine spongiform encephalopathy, variant Creutzfeldt-Jakob. Recognition of virus. How long viruses have been within our 1500 BC, leg deformities indicative of poliomyelitis and bookmarks indicative of a small book. Navarro Silta ruled Egypt from 1200 to 1193 BC, when he died suddenly at the age of about 20. His mummified body lay undisturbed in his tomb in Valley of Kings until 1905, when the tomb is excavated. The mummy shows his leg was withered, his foot was rigidly extended like a horse hoof. The pharaoh Ramses V, who died in 1196 BC, is believed to have succumbed to smallpox compared to the pustular lesion on his face and his lips. During the age, all infectious agents were considered to be virus until Koch developed pure culture technique, which allowed the separation and growth. Bacteria were purified and established as disease-causing agents, and then became possible to distinguish them from filterable agents, those able to pass through a special filter designed to prevent the passage of bacteria. And this filterable disease, the coronavirus in 1898, yellow fever, flavivirus in 1900, Rau sarcoma virus, and oncogenic retrovirus in viral disease have played a major role in human history over whatever time scale we choose to explore. Over the past 1,000 years, smallpox and measles were brought to North and South America by early European explorers and conquerors. These diseases, for which the Native American population has no acquired partial immunity, killed large fractions of the population and were a major factor in the decimation of these societies. As the century entered its final 20 years, new 100 lethal virus HIV spread rapidly around the world via body fluid transmission. Over the past 100 years, a newly emerged strain of influenza killed 20 million people in 1980 and 1990 in the immediate aftermath of World War. And this is picture displayed. A decade later, polio became one of the most feared infectious children and young adults, including Franklin Roosevelt, the US president, throughout Depression and World War II. The global HIV epidemic continues. Sporadic cases and outbreaks in human of some non-human host virus such as Ebola and Hanta raise the concern about future epidemics by other viruses in new century. Coronavirus, severe acute respiratory disease SARS, is a contagious and sometimes fatal respiratory illness. SARS first appeared in China in November 2200. Within a few months, SARS spread worldwide, carried by unsuspecting travelers. SARS showed how quickly infection can spread in a highly mobile and interconnected coronavirus disease. In 2009, COVID-19 is a contagious disease caused by virus, severe acute respiratory syndrome. Coronavirus 2, the first known case, was identified in Wuhan, China in December 2019. The disease quickly spread worldwide, resulting in COVID-19 pandemic. The discipline of the virology, the study of virology inherent, involve a merging together of what has traditionally been thought of as two separate kinds of science, basic and applied science. We want to figure out how viruses are transmitted, how they replicate, and how the host organism respond. We also want to figure out how to prevent transmission, how to interfere with the virus application, how to confer immunity on the host. The applied follows form and is dependent upon the basic in virology, as it is studied today, is therefore an outgrowth of both the infectious disease and the molecular biology. Infectious disease because of the recognition of the viral pathogens, molecular biology because of the usefulness of the virus as probes of cells, and molecular biology and metabolism, and as a vector with a strong potential of genital risk. This is the primary mouse primary spleen cell transduced with a green fluorescent protein, retrovirus vector. Animal viruses classified the DNA viruses, and RNA viral genome. According to classification according viral genome, the nucleic acid, DNA, there is either single-stranded RNA virus or double-stranded DNA viruses, and RNA double-stranded, or either double-stranded or single-stranded. The single-stranded either positive sense or negative sense. The RNA virus should transform to DNA before integrated onto the genome of the host by reverse transcriptase. Single-stranded RNA genomes, positive sense or negative sense or antisense. The positive sense directly translated to amino acids. Negative sense must be reversed to the positive sense before translated to the amino acids. Single-stranded RNA genome, the virus with single-stranded DNA genome, circoviridae and porcine circovirus, parvoviridae, canine parvo, Pylopodenia virus, porcine parvovirus, plus since RNA virus, and this is a mixture of some viruses. And this is a minus sense RNA viruses. The virus with the double-stranded DNA genome, Parvoviridae, papillomavirus, and adenoviruses, and the herpes viridae, bovine herpes virus, equine herpes virus, porcine cytomegalovirus virus, malignant catarrhal fever, poxviridae, pox viruses, African swine fever virus, African swine fever. And this is plus since RNA viruses, some pictures, single-stranded RNA genome. The virus with double-stranded RNA genome, Rhinophairidae, rotavirus, virus, African horse sickness, Werner-viridae, infectious dorsal disease, chicken, infectious pancreatic necrosis, salmonid fish, single-stranded RNA. Virus with double-stranded RNA genome, Rhinophairidae, rotavirus, virus, African horse sickness, Werner-viridae, infectious dorsal disease, infectious pancreatic. Virus with negative sense RNA genome, Orthomyxoviridae, influenza virus, Paramyxoviridae, parainfluenza virus, canine Epstein-Barr, Hendra, Carniva virus, respiratory syncytial virus, Rhabdoviridae, rabies virus, vesicular stomatitis, Flaviviridae, Ebola virus, Boniaviridae, Hantan virus, virus with reverse transcriptase, Retroviridae, feline leukemia virus, feline bovine immunodeficiency virus, bovine and avian leukoplakia.